this video clip, I'm going to introduce to you another method for sampling populations. And this is a really important method because it is the one that allows us to sample mobile species. So anything that can move, we need to use this mark, release, recapture method to make an estimate of the population size. Now the exam board are really keen on the AO1 of this, so you've been able to describe the method in detail because there's lots of things people often forget to mention in their method. But they're also at AO2 really keen on getting you to explore the assumptions the method makes and how that might mean limitations in terms of the estimations that you make, either an over or underestimate, depending on the scenario. So that's applying your knowledge of the assumptions. So in this video, we want to introduce you to the concept so we can spend some more time in class applying and practicing the exam questions. So you need to turn to your keynotes, page 9, where you'll find this information, and I'm going to take you through. Okay, so our method to start. Okay, so you're following this on page 9 in your keynotes. So the first thing we've got to do is we've got to capture a sample of animals and count them. Okay, now it may be that the exam board want you to expand on how you would capture your particular species of animal depending on what type they are. Now, you're not going to have an extensive knowledge of different capture techniques. So we're talking about something quite simple. So you can see in your keynotes, it might be that you think you would use a net or a trap to capture the organism. It wouldn't be important that capture is done in a way which doesn't harm the organism and doesn't harm other organisms. Okay, so this is my forest, and in the forest live lots and lots and lots of mice. So I'm going to show you how this would work. I'm going to take a sample of my mice. Okay, to be representative, I want a big sample. So these will be caught. Next, the animals are marked. Okay, now again, this is where you would expand the detail of your answer. So as you can see in your keynotes, it's really important that you suggest a way that the organisms would be marked that's appropriate to that species. So for example, like a little woodlouse. There's my woodlouse, very good diagram of woodlouse. If we were to mark it on its sort of top with fluorescent yellow paint, that would probably highlight it to predators and so make it more vulnerable. Okay, so we would choose not to mark it in a way like that. So the detail of how you mark it might be what is the mark point on the exam script. So if you were sampling woodlice, you would get your mark not for just saying that you mark them, but for maybe saying you mark them with a non-toxic paint on the underside where that would not be seen by predators. That way, your mark doesn't affect the survival of your species that you're sampling. Okay, for bird species, it's often more appropriate to mark them by placing, so this is a bird in case you can't tell, placing a ring around the ankle. Okay, so depending on the species, you'll choose a way to mark them which is safe, not toxic, and doesn't cause them any harm by making them more visible to predators. So I'm now going to mark my first capture. So I've counted them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 mice. And now I'm going to place a mark on each of them. Okay, once you've done that, you're going to release them back, and the release has to be back into the environment from which you caught them, so at the same sample site as you originally sampled. Okay, now the third step is really important. Okay, so this entire methodology works because we're making an assumption. We're calculating the population size, assuming that the proportion of marked individuals that we will go on to capture in our second sample will be the same as the proportion of marked would be in the whole population. Okay, so that is how this methodology works. So unless we allow the time between our two samples for the organisms to disperse themselves equally, remember these are our marked individuals, throughout the population, then we won't get a good estimate. Okay, so it's really important that the way you write it implies to the examiner that you've given enough time for them to disperse equally back into the population. Now in your keynotes we said leave enough time for them to disperse into the population. You might want to add the word equal in just to make sure that it's really clear to the examiner. Okay, so once they have put them back where we caught them, the important thing is that they get redistributed equally within the population. So we want to make sure that the mice get equally distributed. Now that is usually about giving them enough time. Okay, and so our fourth mark point in the method is what we do with our second capture. Okay, so this is our second capture. We take another sample from that same population. And this time we've two counts that we need to make. We've got to count the number of marked individuals in that second sample and the number of unmarked. Okay, and the reason for this is because we are going to be using those numbers to calculate an estimated population size, and we're assuming that the proportion of marked individuals in the second sample is the same proportion as marked animals in the whole population. So it's kind of the ratio of the number of marked to unmarked that we're using to make our estimate. Okay, so now I'm going to resample. Okay, so taking my sample, I'm going to count how many I have got that have a mark and how many are without a mark. Okay, so we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 mice. Of that, 1 has a mark, 2 has a mark, and 3 have a mark. Okay, and the final mark point, which people always forget to write, is the how. So how do we use those numbers to come up with an estimate? Okay, you need to learn this formula. Okay, so the population size, and remember this is an estimate of population size because we're not counting all the animals, we're just taking 2 samples. It's calculated in the following way. The number of individuals caught in the first sample times the total number caught in the second sample goes on the top line, and then that is divided by the number of marked individuals in the second capture. Okay, so in the example of our mice then, let's apply our formula. Okay, you can stop the video here and give it a go. So my first capture, so my N1, that was 13, and I'm going to be timesing that by the total number in my second capture. Now this is often how the examiner gives the data because I could accidentally now make a bit of a boo-boo, and some of you might have done that. Remember, the 2n is the total number caught in the second sample. So that's my marked and unmarked, so that'll be 15. Okay, and then that's divided by my marked individuals in the second sample, which is 3. So let's have a go at my estimate of my box forest. Okay, so 13 times 15 divided by 3. So I've got an estimate of 65 mice. Okay, so if we think then about how this method works, there's a number of things that you might need to add as additional information. Okay, so it may be that the examiner's asked you to think about this in a particular context, and there may be some follow-up AO3 questions as well. Okay, so our AO1 is to learn off by heart that method, okay, which we'll test. But we also now need to be able to apply it. Okay, so what I'd like you to think about now is applying this method. So how would you estimate the ladybird population size in a given area? Okay, I want you to think about the sampling method, which we've just introduced. So how would you describe that to the examiner? How would you make it specific to ladybirds? Okay, how would you ensure then, so extra bits of detail you could add if you're designing a method, a specific method. So we know that we're using the mark, release, recapture methodology, but we've also, as good scientists, got to make sure that we're unbiased and that the estimate we make is representative or reliable. So what things in terms of our sampling can we do to ensure that? Okay, so have a go and then we'll see. Okay, so how would that look? Okay, so ladybirds, well, first mark point, you've got to capture them. Okay, now a netting technique might be fine for this, or some sort of pitfall trap. Okay, so let me show you some of the different methodologies that you can use to sample different organisms, things you might be familiar with. So this is a Longman trap, so that's a humane small animal trap which traps things like mice without causing them any harm. So it's got a little trigger here, the mouse goes in and the door closes behind it. So that'd be appropriate for things like small mammals, but you don't need to know any detail about that. You would just be talking about using traps with things like mice. Okay, for things like fish, you might be using, or things that live at the bottom of a lake or stream, you might be using a net technique. Okay, so picking the appropriate sample method for a ladybird might be a net like this one here. So you might be netting an area. You might be collecting all of them using a pooter. You might be doing a branch shake and shaking them off a branch, or you might use a trap. Now this is a moth trap, but that kind of moth trap, we've got similar pitfall traps where you bury like a beaker and then you put a bit of stone over the top like that. You might have used a trap like this to trap insects, and the insects walk along and fall into the trap and get stuck, and you can collect them that way. So there's loads of different sampling methods. Now, the examiner is not going to expect you to know all the detail of all of these, but they may describe one to you, okay? Or if you use the words net and trap, they cover you for most things. So you would net insects and fish and trap small mammals. So we need to give a sort of appropriate sampling method. We need to make sure it's unbiased. Okay, so in exactly the same way we placed our quadrats in a random place, we took a quadrat measurement at a random coordinate, you need to think of a way of making sure that your sampling is random in your first and second sample of your site. Okay, so that's really important. There's different ways of doing that. I would suggest we just use a similar way. You can do sort of coordinates if they ever ask you about it. as you would like with your quadrats, an x coordinate, a y coordinate, randomly selected from the calculator, and instead of placing the quadrat here, that would be where you'd place your trap and leave that overnight to capture your species. The final thing I want you to think about is making it representative and reliable. Okay, so when we say first capture, it wouldn't be representative and reliable if you only set one trap and had one sample, or used one sort of net at one site in that first capture, and then the same in the second capture, because that wouldn't be enough samples. Okay, so of course you will, just like all science methodologies, you will make sure that you do many samples within your first capture and your second capture, if you like. So many traps or many nets. And there may be additional mark points for expressing this to the examiner, so when you're given a detailed method. So how would that now look? Okay, so we're going to do point 1. We're going to capture the ladybirds. We're going to use a net to capture them. We're going to make sure that in our sampling we sample randomly a large number of sites. And now I've just improved my first mark point to make sure I've got AO1 application in there. Okay. So, other things the examiner likes to do at AO3 is just to get you to think about why your estimate might not be very good. Now, we're going to move on to the assumptions in a second, but the other thing you might want to consider are limitations to your practical. So, things with ladybirds, just to throw out there, ladybirds at different times of the year are in different times in their life cycle. So, if I went out to collect ladybird numbers in July and June, and I was counting adults, I would get a very, very small number. Now, would that be a true representative sample of the ladybird population? No. So I do need to know my species, and I need to know things like if the adults will be available to count in that month that I'm sampling. Equally, the numbers might fluctuate at different times of the year. So it might be that to overcome these limitations, I would do that estimate every month throughout the year, the same area, and do an average or something like that. Other limitations, there's lots of different species of ladybirds. Which ones am I actually sampling? Can I tell them apart? Okay, so now we've taken that first sample and we've added that extra detail to apply this methodology. We've still got to cover all the rest of the mark points. Okay, so we still now need to talk about marking our ladybirds. Again, you need to be specific here, because we want to talk about marking our ladybirds in a way which means that the mark is not toxic and it doesn't increase their susceptibility to predation. Okay, so that's really important. Once we've marked the ladybirds, again we're going to release them back into the same area that they were sampled from, and we're going to give an adequate amount of time to enable them to distribute equally. Okay, we're going to come on to some of the AO3 assumptions, the assumptions of this methodology in a moment. And when you say time, enough time is given for the ladybirds to distribute equally, you may also add in at that point, but however not enough time, that their numbers would change due to births, deaths or migration. And that's a point I'll come back to in a moment. Then we would have to talk about doing our second capture. Okay, so we can take a second capture of our ladybirds using the same methodology. It'd still be random, and we would still use the same way in which we capture them. This time we're going to count all the marked individuals and the unmarked individuals in the sample. Okay, and we will always get our 5th mark point for stating the mark release recapture formula. So that is that the number of individuals in our first capture times by the number of individuals in our second capture divided by the number of marked individuals in the second capture is our estimate, so equal to our estimated population size. Okay, so to answer that question, to apply our AO1 knowledge of the methodology to our ladybird example, those are the additional points we would put in. Okay, so now thinking about our assumptions. So final AO1s, these are learn off by heart, like your method, but you do need to be ready to apply them. Okay, so first of all, we're assuming that the mark does not harm the individual in any way and does not make them susceptible to any predators. Now, they are always the same mark point, so don't give them as one mark point. Oh, sorry, it's two separate mark points. They're only one mark point together. The other thing that we say is that you're assuming that the time between the two samples, the first capture and the second capture, there will be no change in the population. So that's really important. If the examiner indicates that's a long time between the two samples, like for example a year, then of course that assumption has been broken because the population size will probably change in that time. So there must be no births, no deaths, no migration. Again, that's one mark point. The next one is that there's been enough time between the two samples for them to fully redistribute themselves before the next sample is taken. So that's one mark point. And finally, I'll talk to you about that one in a second. Okay, so on page 9 at the bottom of the page, so there's several important considerations. Okay, now remember this formula is based on the ratio of marked to unmarked individuals. So anything that would affect that ratio would affect your estimate. So population size doesn't change between the two capture times. So there are no births, no deaths, no migration. Those first 3 bullet points we've said then are one mark point here. So don't say them as separate mark points. It's one mark point. Marking doesn't affect the survival. So this is doesn't make them more susceptible to predators or toxic. That is this first set here. That's one mark point. So don't say them as separate. And the sufficient time is our third mark point. So you need to learn those 3 off by heart. That's your AO1. We do need to watch out for them at AO3 though. Okay, so this is what we're going to be practicing in class. So when you know your assumptions, we will be asking you to apply them to methods that other scientists have done to see what's wrong with the method, so what the limitations of the method would be. And if they break assumptions, then so the assumptions that we're having here are broken by something that happens, so the mark does make them more susceptible, then that would be a limitation of the practical. It will cause an over or underestimation. And I'll show you how with one example. Okay, so pause here and have a go at this one. Okay, so calculating the estimated size of the ladybird population, showing you working out. So estimated size of the population, there were 100 trapped in the first sample. They were marked and released. A week later, second sample, we got 30 that were marked and 15 that weren't. So 45 divided by the number that were marked, which is 30. Okay, so 150 ladybirds. Explain why it's important that the sample contains as many ladybirds as possible. So those 2 samples, so that was about making it reliable and representative. Okay, so that's a large number, representative and reliable. Okay, so what I want us to do now is to think about applying our assumptions simply to AO3. So the examiner tells us another scientist has used the mark, release, recapture method, and the following has happened. So when they marked their animals, there was a toxic substance used. What impact would that have on the population estimate? Well, let's have a think. Okay, so if we use the same data we just used, in the first instance we caught 100 ladybirds and we marked them with a toxic substance. So in my second capture, I might still capture the same number of ladybirds, 45, but if they had that mark on them, they would have died or would be very likely to die. So that means there wouldn't be very many marked individuals left. So this number at the bottom wouldn't be 30, it might be 0 or 1. Let's make it 0. Okay, so now my estimate has gone up from 150 to 45,000. So because the investigators have broken this assumption, they've got an overestimation in their population. Could you have a go at the next one, okay, and see what impact you think it would have on their estimation? So scientists have used the mark, release, recapture method to estimate the population of ladybirds or mice. Mice would be better in this example. Okay, now the population has developed many trap happy individuals. Now this sometimes happens with small mammals, and it's when the trap's such a nice place to be, there's food, it's warm, that once they've experienced being in a trap, they seek them out and are much more likely to be second trapped, if you like. Okay, so how would that affect our estimate? So let's think about the same numbers but with mice. So in our first sample we'd still have 100 mice. Now of those 100 mice, they've been trapped. They enjoyed the experience. Okay, so we might still only have the same number in our second sample, but of those, they're likely to have been mice that we have captured before. Remember, those 100 have been marked. So if I divide that, then I'll be dividing it by a bigger number. I'll be much more likely to capture the mice that had a mark that were caught in the first capture because they like being trapped. Okay, so now you can see the estimate has been dropped significantly lower. So we've got 100 mice. Okay, so the examiner can give you different scenarios and ask you how that would affect the estimate. My advice is always to students to put some numbers, pretend numbers in your mark, release, recapture formula, and then imagine what would happen to them in the scenario. Okay, so you can pause here and have a go at C and D, but I'll talk you through them. Okay, so what about if the mark washes off? So we'll start with 100 still, and we've still got 45 in the second sample, so we'll just start with the same numbers. But the mark washes off. So of those 100 that have been marked, the majority of those marks have gone. So now we'll be, instead of 30, it would drop. So maybe there's only 5 left with a mark. Okay, again, we should be able to give whether this is an over or underestimate. So it's going to be an overestimate. Okay. And migration, so this is the unmarked individuals moving into the area. Okay, so remember this one's a bit harder to think about because it's a ratio And what's happening is that your ratio is going to be affected by the influx of unmarked individuals between the two samples. So as they migrate into the area, it means that there will be fewer marked individuals in that second capture, because of course it's a ratio. So that one's a bit harder to think about. So let's think now we've got maybe 2. Okay, so again we're going to get an overestimation, a much bigger number than, because we're breaking an assumption. So if any of the assumptions are broken, it would lead to either an under or overestimate of your sample, your population size. And what we want to be able to do in our practice now is to make sure that at AO3 you can decipher the impact yourself of different breaks to assumptions. Okay. So have a go at this one, and then we'll review this one in class. Okay, so explain where the animals are marked and released. It's important that the mark is not particularly visible, not toxic, and give me the 4 assumptions. Okay, so I'm going to come back to this in class, but not particularly visible, so that predators are not more likely to eat the marked individuals, which would lead to an overestimation, as well as being not ethical. Not being toxic, again, if it was toxic, it would kill the marked individuals and lead to an overestimation, so also being unethical. And the 4 assumptions, okay, remember, if you're going to do the assumptions, really, I've narrowed it down to 3 more than 4 that are 3 good mark points. They used to give all these separate bullet points as separate assumptions, but now they kind of group them together in the mark scheme. So that the population doesn't change between the 2 capture times, no births, no deaths, and no migration. That's 1 mark point really. That the marking does not affect survival, so either making the marked animals more susceptible to predators or being toxic. And then finally, that there's sufficient time between the first capture and the second capture that the first capture organisms that have been marked equally distribute throughout the population. Okay, so we've got another couple of things that you need to add to your AO1 bank for this. So there is a little grey box to read through on page 10 about ethics. So it does mention in our specification that you should think about the ethics of sampling, although I've never seen an exam question yet about it. Doesn't mean it won't happen. So just think logically about how your sampling should not impact the ecosystem locally, so that the idea that the individuals that are captured are unharmed and then returned to the same habitat with no damage to the habitat. And that any movement or sampling doesn't damage the habitat or affect the distribution of other species. So have a read through that. And then finally, with all of this, it's about measuring populations, investigating populations. Now we do that in science so that we can look to the impact of something. So you might be monitoring the population of bees, native bee species in the UK across different sites. Now that would be an ongoing process. We might be doing those repeatedly over years and years and years and years. So often when you're given these kind of exam questions, there's some statistics that is brought into it. So remember that link. If you're asked how the scientists would see if the change in population that they've observed over a number of years or between 2 different sites is significant, of course you're going to suggest using a stats test. So remember to bring in stats tests as well, as we will in class. Okay, right, that's it for this video.